Every second, a million or so cells die in our body. The vast majority of those cell deaths are through a pathway called the mitochondrial pathway of apoptosis. The mitochondrial pathway is remarkable in that, we usually think about mitochondria as being to provide energy for the cell and provide other vital functions for the cell. In this case, though, the mitochondria are really the suicide capsule of death. What happens during the mitochondrial pathway of apoptosis is that the outer membrane of the mitochondria suddenly becomes permeable, a process we call mitochondrial outer membrane permeabilization, or MOMP. The process of MOMP and how it's controlled is really another key to understanding apoptosis. But once it's occurred, the chemical process of apoptosis proceeds, and that's because molecules from the mitochondrial intermembrane space now gain access to the cytosol in the cell, where they engage other molecules. The most important molecule involved in this process is plain old cytochrome C. APAF1 lives as a monomer in all of our cells, in the cytosol of every cell in our body. APAF1 is inactive in that form. When cytochrome C binds to APAF1, it undergoes a conformational change, and it now forms an oligomer, which is an elegant seven-spoked structure, the center of which is a region that binds and recruits the next molecule in the pathway. That molecule is one of the caspases, caspase 9. When caspase 9 is brought to the center of the APAF1 oligomer, which we call an apoptosome, that now dimerizes the caspase 9 and activates it. Now, caspase 9 has only two substrates that we know about or care about, and those are caspases 3 and 7. That cleavage activates the executioner caspase, and now apoptosis proceeds.